All right. Good afternoon, everyone. My name is Mark Nardone. I'm director and co-founder of Bio-Trac. Welcome to our Thursday weekly webinar series. Those who are not familiar with our program, Bio-Trac offers in-person, hands-on, and remote training workshops for research scientists. These workshops, which are team taught by active researchers, focus on the latest relevant methods, techniques that are used in laboratory research. Currently, we have workshops that focus on, but not limited to, gene editing with CRISPR, exosomes, hepatocytes, stem cells, iPSCs, flow cytometry, 3D cell culture, single cell RNA-seq, and other areas of NGS. Along with our hands-on training workshops, we continue to serve the scientific community through custom design training programs, biopanel networking events, weekly webinar series, symposiums. We host government and industry retreats, as well as other outreach initiatives, such as the supporter of the Maryland Science Olympiad. Today, it's my pleasure to introduce to you Dr. Bernard Heyman. Dr. Heyman is part of the NIH community for the past 20 years, currently as a staff scientist with the NIMS Laboratory of Structural Biology Research. He graduated from Purdue University, receiving his PhD in biochemistry, returned to Switzerland, where he worked as a postdoctoral fellow with Dr. Andreas Engel on the red blood cell aquaporin in collaboration with Dr. Peter Agre, a Nobel laureate. At that time, he began to develop a software package, BSOF, for the processing of structural biology data. In 2000, Dr. Heyman joined the NIMS as a postdoctoral fellow in the laboratory of Dr. Alistair Sieben, studying the maturation of herpes simplex type 1 capsid. After a brief period as a staff scientist at Caltech, he returned back to NIMS as a research fellow and later as a staff scientist with Dr. Steven, continuing to work on determining the structures of biomolecules and developing the BSOF software program. So without further ado, Dr. Bernard Heyman, the opportunities of cryo-electron microscopy and biomedical research. Bernard, they're all yours.
Thank you, Mark. Thank you for that introduction. Today, I want to give you sort of an overview of the kinds of things that we are doing in CryoEm for the biomedical research community. This is going to be a very superficial talk, and I'll only touch on a few things. And the reason for that is that electron microscopy is an enormous field that started less than 100 years ago with the invention of the electron microscope. And in this picture, you see the development over the years of what we call three-dimensional electron microscopy, with the first 3D structures being determined in the 1960s. And with some progress, and as you see the dates here, that sort of accelerates. So I've been involved in the field for about 30 years, and it used to be a very small community, but now with the development of much better microscopes and much better techniques, the whole field has exploded, as you can see in this graph of the 3D reconstructions deposited in electron microscopy data bank. You can see that even a few years ago, there were only a few in the data bank, and it's now exploding exponentially. And that is
for 1 second, Bernard. Right now, we're just seeing your cover slide. It did not advance.
It did not advance. Do you see it now?
No, it's still the opportunities, the title slide.
Okay, it says sharing is paused. I'm not entirely sure why that is.
Okay, let's, how about you unshare and then share again?
Okay, resume share, okay. It's not allowing, oh, here we stop share.
That looks good.
Okay. Okay, so you can see the slide, which has a timeline of cryo-EM development. And as I said, the development started in the 1930s, which is less than 100 years ago. And since it has been exploded, as you can see in this graph of the maps deposited in the EM data bank. Can you see it, Mark?
We can, yes.
Okay, very good. So as I said, electron microscopy covers many techniques. And here I have a listing of the sort of non-cryo techniques. Some of them, that's the traditional embedding and immunolabeling that you typically see in papers. But then there are a very large number of other techniques that one could apply to biological materials to get useful information. And I'm not going to go through the whole list. In a section with cryo, probably the one that people are mostly talking about these days is, or understood under cryo-EM.
Bernard, I'm sorry to interrupt. I'm sorry. We're just seeing the timeline right now. It did not advance.
Okay, it keeps telling me it's paused.
Very strange.
Okay, let me try it a different way. Okay, how about now?
Okay, right, yes, right now we're seeing the list of topics.
Okay, so as I said, there's a very long list of topics that's under of techniques that you can use in electron microscopy, of which the cryo techniques are the more modern techniques that have been developed since Jacques Dubochet and his co-workers developed the methodology to freeze biological samples and for us to exploit the inherent capability of the electron microscope to get to really high resolution. So I'm going to concentrate on a few of the topics under the cryo, some of it the single particle analysis, some of it the tomography, and then I'm going to get into some of the more advanced methodologies that we are trying to develop to look into cells and tissues. And so how can we use electron microscopy? Here is a graph that sort of shows you the distribution of techniques or the length scales that techniques can address. And you see NMR spectroscopy and X-ray crystallography on the very high resolution side, and then on the low resolution side, you have light microscopy. So electron microscopy is actually the technique that can bridge the atomic level to the cellular level. Now, since electron beams are still very destructive and very high energetic, we cannot have samples that's too thick. So our cutoff is roughly around 1 micron. So your sample has to be about 1 micron thick. In the past, we had 1 million volt microscopes that could penetrate thicker samples. But the problem with thicker samples is always that you lose resolution. So by the time that you get to a really thick sample, it's not really that much better than light microscopy. But anyway, electron microscopy, as we are developing it today, is really geared towards bridging this gap between the atomic and the cellular levels of scales. Now, quite recently, we have this beautiful structure of the apoferritin at 1.2 angstroms, where you can actually visualize individual atoms, even individual waters. And so this is an astonishing level of achievement for the electron microscope since its development. On the other side of the spectrum that I just showed you, we just in the meantime, I want to say that this is one of the ways in which we would like to look at drug development is we can now almost replace X-ray crystallography with the ability to actually look at proteins without having to crystallize them and to be able to locate drugs in them. And this is one of the ways that companies like Genentech is now deeply involved in CryoEm for drug development. I'm not going to talk much about drug development, but that's one of the major applications of CryoEm. On the other side is actually looking inside cells and even inside tissues, and actually to be able to locate all of these biological molecules we can look at on an atomic structure or a near atomic structure and locate them within the cell and then see what the relationships are in a structural way between each other and between the membranes and the organelles and so on in the cell. So this is just to give you an idea of the range of scales that we span in electron microscopy. So the way that electron microscopy is used in health research is for the most part on the basic side, although I'm going to show you some things on the clinical side where we look at antibody function and of course drug design that I just mentioned. We're always thinking about ways in which we could potentially use electron microscopy for diagnostic purposes. But because the preparation and the computational side and so on is so involved and complicated, is that we don't really have good diagnostic techniques based on cryo-electron microscopy. But this might change in the future if we have more automated pipelines. So on the basic side, this technique can be used in various ways. The main one is structure determination, just like X-ray crystallography. But then because we are looking at biological molecules sort of within their natural environment, we can actually look at the dynamics. And I'll show you an example of what I did in the past. We can also look at compositional analysis, much easier than you do with X-ray crystallography. You can literally have a sample with a variable composition and you can analyze them and classify them into the different conformations and compositions. And then as I showed you, one could localize these biomolecules within cells. You can look at the dynamics within cells, the distribution, how they interact with other components, and how they interact with the cellular morphology and how they affect cellular morphology. So this is all part of the theme of bridging the gap between atomic structures and cellular structure. As in every technique, one of the hardest things to do is to help prepare your samples for a suitable preparation for electron microscopy. And this goes back to what I said about diagnosis. It's in most cases pretty hard to prepare samples for electron microscopy. Once you get a good sample into electron microscope, it is actually these days relatively easy to image it and to do the data processing. We get better and better software, more automated pipelines. And so eventually you might be able to look at the sample and within days get three-dimensional reconstructions. Now, the other part of after you get the three-dimensional reconstruction is how do you interpret it? And this is where a lot of effort goes into it. On the atomic side, we have all the history of building atomic models into electron densities obtained by X-ray crystallography. And so we can use all of that. And because that is very well developed, that is almost a non-issue these days. On the other side, if we look at cellular structure and bigger structures and so on, we still have a long way to go before we understand how to interpret everything in a way that is easy and fast and give us reasonable answers. At the moment, that kind of interpretation takes a lot of effort. So one of the first things that you want to do when you want to get a sample into an electron microscope is you want to get it onto one of these electron microscopy grids, which typically is a 3 millimeter copper grid with perforations in it. It's typically coated with a layer of plastic onto which you then put your sample. You want to get it into the microscope with a typical sample holder. Some of the modern microscopes have different kinds of holders that is more automated and suitable for automatic loading of grids. To prepare the grids, you need a device that can do plunge freezing. And I'll show you some more about this in a moment. And then eventually you want to end up with a grid, with your sample on the grid. On the leftmost image, you see that a typical grid with the grid bars and everything. In the middle image, you see a close-up where you have a support that have holes in the support. And this is because we don't want to have the background that's associated with the film. So in these holes, you just have a very thin layer of vitreous ice in which your sample is frozen. And here is another close-up within one of those holes with a set of particles. And so this is how the device works. First, you get your sample on the grid over here. You blot the excess liquid from the grid, and it then drops into liquid ethane that's at the temperature of liquid nitrogen. And this freezing process is so fast that the water cannot crystallize because that would damage your specimen. And so we talk about vitreous ice. And so this is the ideal that you want to obtain is a very thin layer of vitreous ice in which your sample is embedded. And this is just frames from a movie, a high-speed movie from the Casas paper, where they looked at what happens and how fast they can get the grid into the liquid ethane and give you an idea of how fast it happens to get vitreous ice rather than crystalline ice. So when you've done this, you get a sample, and this is one of the micrographs that I worked with in my career of the herpes simplex virus. And you see it looks actually very noisy. You can nicely see the capsid particles, but if you have a much smaller protein, it's much harder to see it. And that is one of the things that's always true in cryo-electron microscopy, is that the pictures are very noisy because we have to use low dose. And that is because the electron beam is damaging, and it's a race between obtaining information and destroying your sample. So in studies we've done, the moment you start illuminating your sample with the electron beam, you are damaging it. And in the graph on the right, you see the resolution of reconstructions decreasing as you increase the dose. And these kinds of things have now been dealt with in the modern microscopes. I'm not going to go into much of a detail there, but we handle it much better now so we can get to really high resolution. Okay, so let me start with an example of some of my old work. So in the herpes virus capsid assembly, it starts out with a procapsid, that is this sort of spherical particle with a lot of perforations in the capsid. And then over time, it spontaneously matures to this mature form where all of these holes have closed up. The capsomers here have become more regular. And so we were interested in looking at how this process happens. And so here is a movie that I made many years ago about this transformation. And so here you see the procapsid, and eventually it closes up the gaps in between the capsomers, and form the mature capsid. And this is just an inside view. And so so electron microscopy allows us to actually classify these particles into their different structures and then recreate the timeline of the maturation. And so this is something that you cannot really do with many other techniques at this resolution. And then once you have a structure that you can fit into those maps, you can actually start looking at the dynamics of how these structures come together, how they interact, and so on. So this was done a long time ago, so it's not such high resolution work. And these days we can do it much better than that. But that's just to illustrate to you that the dynamics, we can really access the dynamics through electron microscopy. In another related study, also on the herpes simplex virus, we wanted to do tomography to actually find the so-called portal protein that is mostly on one vertex in the capsid. And so this is an example of tomography where you just tilt the specimen in the microscope, and then eventually you reconstruct and average these capsids, and you then can identify a particular vertex where this portal protein is. And this here I show you the result of that, where we've identified the portal protein on one vertex. You can see the capsid on the outside, the so-called scaffolding protein on the inside. And we can fit an atomic model of the portal protein into this density, and shows us how it's oriented relative to the rest of the capsid and how it's connected to the rest of the capsid. What this also allows us to do is to actually count how many portal proteins there are per capsid. Now, the conventional wisdom was that there was only one per capsid, but this tomography allowed us to calculate it. In some capsids, there are none. In most capsids, there are one, but there are some capsids with 2 or 3. And so again, it's a technique that is, we cannot do the same kind of thing in many other techniques. Okay, so we've done a lot of tomography of viruses because it's just simply easy to purify a virus and throw it in the microscope and make it. When we actually want to look at how these microscopes evolve inside the cells, then it becomes a little bit more difficult, and we have to have other techniques in which we can actually isolate the thin part of the cell, where we can then image it by electron microscopy. And here I just have an example of it on the right side, where people have imaged this flockhouse nodovirus to look at the assembly of the virus in the cell. It actually assembles between the membranes of the mitochondria. And so in different viruses, there are different routes in which the virus assemble. It may assemble in the cytoplasm, may assemble in the nucleus. So every virus has its own assembly pathway. Okay, so I want to say something about the coronavirus, since we're in the middle of the epidemic. And many years ago, one of our co-workers at the NIH, Al Kapikian, looked at the coagulation of coronaviruses by convalescent serum, or the antibodies in convalescent serum. And in the right-hand side, you see a negative stain electron micrograph where they've demonstrated that the convalescent serum actually coagulated these viruses. And this is a mechanism in the body for the immune system to rid the body of the viruses. So since we're interested in how antibodies bind to viruses, we've done studies like on this hepatitis B virus, where we've characterized the binding of antibody to the spike of the virus. And here you see examples of 6 different monoclonal antibodies. and how they bind to the virus capsid. This is important if you want to design better antibodies, or if you design better antigens to elicit antibodies in the use of vaccines. We can also look at how antibodies from convalescent serum bind to a capsid like hepatitis B virus. And because this is now a mixture of antibodies, it sort of helps us to characterize how antibodies in general bind to it, and where it agrees with our studies of monoclonal antibodies. So this is part of the application of electron microscopy, where we can actually characterize these antibodies and see if there's any way in which we can improve the antigenic nature of vaccines. To come back to the coronavirus, now, so in the top panel, you see the coronavirus spike protein with some of them complex to the ACE2 receptor. And this was the original SARS, the SARS that broke out in 2003. And people have been working on this and solving the structure of the spike protein. So by the time that the pandemic hit, the beginning of last year, people just switched over to SARS-2, and the spike protein, the structure of the spike protein was determined within a month of people knowing that the virus is coming out of China. So that just gives you an idea of how rapidly we can respond in CryoEm to new developments. Now, we can also use it, of course, to then look at antibodies binding to the spike protein, and how effective that is in neutralizing the virus. Other things that have happened in the meantime, because of the focus on the epidemic, is that people have started looking at some of the other proteins. And here is, for instance, the 3A ion channel, which is implicated in inflammation. So this might be one of the proteins that's involved in some of the severe consequences of infection by the virus. And then, of course, you can study the virus by electron tomography. And so here is a study, and this is actually also, I think some time ago, where they then look at the orientation of the spikes on the surface of the virus. You can see in what conformations the spikes are, and you can actually see the conformation of the post-fusion spike, which looks very different from the pre-fusion spike. So this is the conformation that then would insert into the host cell membrane and cause fusion. So again, if you know how this works, you can potentially design drugs to interfere with this transformation from this point to this point. And then you can, with tomography, you can also look what goes on the inside of the virus. And so what they found are these sort of globules of the ribonucleoprotein. So this is the RNA coated with a specific protein, the N protein. And again, the more we know about the virus, the more we can try to develop antivirals. Now, eventually, we want to see how these proteins functions in vivo. And so if you are lucky, the structure that you're interested in might be isolatable, as in this case. This is the photoreceptor rod outer segment. And in this case, it's very easy to prepare outer segments. You purify or isolate the retina. You remove the RPE on the outside of the retina, and then you shake it a little bit, or there are other techniques to get the outer segment of the rod, and then you can freeze it and study it. And this is a study where they've isolated the outer segments, and this is the cilium, which connects the outer and inner segments. And then you can get the 3D impression of what this looks like and start identifying proteins that you're interested in associated with the psyllium. Now, one of the things to prepare thin samples is cryosectioning. And this is similar to traditional sectioning, except that you have to work at liquid nitrogen temperatures, which of course complicates the whole process, and it's much more tricky. But here you see an example of what we've been involved in. And then once you've sectioned a cryo sample, you get this ribbon of sections that you can then lay across the grid. And here's what it looks like in a microscope. And so here you see the sections across the grid. This is sections coming off a retina. So this is actually trying to look in the tissue itself. And here's a couple of images that we got out of this. On the left, you see an image of the outer segment. The black dots are gold fiducial markers that we intended to use for tomography. But you see the discs that contain the rhodopsin in the outer segments of the rod segments. And on the right, you see an axon of a ganglion cell in the retina. And you can very nicely see these microtubules. You see the protofilaments in the microtubules. And so this gives us the information that the resolution here is about 5 nanometers. So we can distinguish features that's about the size of 5 nanometers. Now, another technique to actually prepare a thin sample is the so-called focused ion beam scanning electron microscope. So in the typical example here is you grow your cells on electron microscopy grid. And then you take it to the CryoEm, which is this machine here in the middle. It has 2 beams. The one is an electron beam for the scanning electron microscopy part of it. And the other one is an ion beam, typically gallium ions, that you can use to actually mill away parts of your sample. And so in the next image, you can see 2 samples. One is yeast, one is cyanobacterial. On the left, you see a SEM image of the 2 samples, where you can see the cells, the yeast cells at the top, and the cyanobacterium, which is sort of a filamentous bacterium at the bottom. Then what you do is you mill away most of it, so you end up with a very thin specimen, typically on the order of about 100 nanometers thick. And on the right, you see what it looks like in an SEM image after you've milled away most of it. Now, this is a relatively slow process, so this may take hours to do this. And so this is this goes into the complications of trying to get the really thin sample going through cells or a tissue. But then if you hit the right spot and the right place, you can get these beautiful images of what goes on inside a cell. And so here you see an example of inside a cell where in the middle you have the nuclear envelope, the 2 membranes of the nuclear envelope. Some places you can see the nuclear pore complex. On the inside, you've got microtubules. And on the outside, you've got the ribosomes and mitochondria and so on. And if you look at the microtubules, you can actually see the protofilaments of the microtubules as well. So this is one example of getting into a cell and actually start interpreting it in a way that you can get down to the molecular level. Another technique is so-called cryocorrelative light electron microscopy, that if you have a fluorescently tagged protein or any fluorescent tags, you can actually combine light and electron microscopy. You can identify where your protein is on the grid in a cryo light microscope. And then you can target in on those areas, and you can do electron microscopy on it and see the structures associated with that fluorescent signal. And in this particular case, they were interested in these ribosomes that's decorating these vesicles. Here you see an indication of a mitochondrion. And then once you have these tomograms, you can extract these things, these densities associated with the ribosomes, and you can do subtomogram averaging, and then you can relate it to the atomic structure. And so you go from what's happening inside cells right down to atomic structure. And you can see how these ribosomes relate to each other, and maybe they form higher order complexes and so on. So this is just to give you a quick idea of what we can do with electron microscopy these days. And I think there are exciting developments ahead where we can actually characterize much more of cells, trafficking through cells, where proteins interact, larger assemblies of proteins, like kinetochores, like the spindles in mitosis, etc. So there's an enormous range of things that we can still get into, and I think we're going to work on that for many, many more years. Thank you very much.
Bernard, thank you so much for a wonderful presentation, wonderful figures and very interesting and timely, obviously. I ask that everyone submit questions in the chat and we can address those. Bernard, we have one already. It's from Felipe. Tubulin has a C-terminal tails that protrude out of the microtubule outer surface. Can they be resolved?
It would be hard to resolve them inside a cell because you have so much material associated with it. But if you have just the microtubules isolated, then yes, higher resolution reconstructions could potentially resolve those. The question there becomes the flexibility of those extensions. If they're very flexible, it becomes harder to visualize them because then they sort of smeared out. It's the same as we get in X-ray crystallography, where if you have very flexible regions, they typically don't appear in the maps. And so that's the main question. But in principle, if those tails are rigid in some way, then yes, we can reconstruct those.
Brian follows that question with, does X-ray crystallography have any role left in your brave new world?
That is a good question, and it is sort of a loaded question. What is certain is that electron microscopy is probably going to become as big as X-ray crystallography, if not bigger. We've seen the other technique that people have been using is NMR for structure determination. And we've seen a downturn in the number of NMR structures being deposited in the PDB. So I don't know, in the long run, maybe X-ray crystallography will go the same way. But at the moment, it is still an enormously useful technique, and people are going to continue using it. I must say a lot of X-ray crystallographers are actually switching over to EM as well now.
Very good. We have a question from Tommy White. If we want to pursue experiments like this, are the resources available? I don't know if they're at the NIH or not.
I hope so. I do know that the number of microscopes that a company like Thermo Fisher are producing is expanding enormously. So there's going to be many, many microscopes available in the world. If you're talking about NIH specifically, there are the regional centers that the NIH has set up that's available for people to, contact and make proposals to. I'm not involved in any of the decisions around these things, so I can't really tell you how available that would be.
Very good. All right, that's all the questions that we have. Again, Bernard, thank you so much for a great presentation.